Hi everyone, it's a pleasure to be with you today. So I'm Nathan Stockman from Eurosafe company, and I will speak today about your cosmetics, their safety and efficacy testing with Eurosafe. So if my slide is going on, no, okay, like this. Eurosafe company is a contract research organization, member of BIOPREDIC group, and our main goal is to provide chemical, biological, and computational solutions for a safer and healthier world. So, for myself, I'm Nathan Stockman. I'm a regulatory toxicologist at Eurosafe, and you have here on the slide my email address if you need to ask questions after this webinar. And first of all, I would like to speak about the life cycle of your cosmetic, because Eurosafe is able to help you at each product development step. So for the ingredient field, we're able to advise you on hazard testing, on toxicological profiling, but also for your finished product on risk testing, in vitro or on volunteers, on safety regulatory, of course, for European safety, but also we'll look for evolution and updates about the regulation itself, or about the toxicological profile and toxicological data on ingredients or on products. So for ingredient testing, we're able to work on the hazard identification, on the in silico modeling if needed, on the cutaneous passage, and also on the efficacy. Yuri from Sala are going to talk to you about that this afternoon. And combining this test with all the literature research, we're able to complete the toxicological profile of every ingredient in the finished product. So we already have more than 3,000 ingredients in our own database. So it's a huge number of ingredients, and it's increasing every day. And for a finished product, beginning for after the ingredient testing, we're able to perform toxicological pre-assessment directly on your formula to check the acceptability, the regulatory acceptability of your formula at an early stage of development, just to not congesting other tests if your formula is not okay. We also perform transcutaneous passage to use real data for the margin of safety calculation. After that, we have the tolerance, mucous membrane or cutaneous tolerance, depending on the product. Of course, for validating the product tolerance, we perform that in vitro or on volunteers. The efficacy, of course, to support your claims, again, in vitro or with the clinical testing, the safety assessment to ensure the safety in full compliance with the regulation, and as I said, the evolution and updates to stay safe during all the product life after placing the product on the market. So with ingredient data, product evolution, regulatory change, and everything. Just to give you an overview of the regulatory field. So as I say, we have the pre-assessment on the left side, the safety tests, the CPSR, the cosmetic product safety report. It's all the European field of the regulatory toxicology. Just to remember that we perform all things need to be included in the PIF, so-called the product information file, the central file of your product in the regulatory field. And we assessed more than 1,000 formula per year. So we're able to perform all the regulatory data. Speaking of tests now, we are good laboratory practices certificated. So here you have, on the right side, you have the agreement for the French authority, medicine authority, the ANSM. And we expected our next inspection of the authority later this year, on December. Here you can see our quality process. We apply this process on each of our in vitro tests. So I will not enter into detail here, but just to show you that everything is under control to be compliant with the GLP certification. So we have a full process for that. Here you have the different tests we can perform. On the left side, you have the ocular and mucosal tolerance tests, the natural red really, the head cam, and also the test on reconstructed tissue, ocular, gingival, vaginal. For each test, you have an idea of the number of products we have already tested, so a huge number of products. And on the right side, you have the skin tolerance tested. Again, at the bottom, the test on reconstructed tissue, the cutaneous tissue, and also the phototoxicity test. And here you have also the PREDIC skin test, the tolerance test directly on a skin disc. So on a skin explant, we can perform a tolerance test with your product or with your ingredient. And this test is performed with a skin tissue provided by BIOPREDIC, and pioneered by BIOPREDIC in France, another member of the WePredict group, and for you in Korea is came from distributed by Coma Biotech. So we can have different products, wall skin, the full thickness skins, dermatose skin, above it. All this the skin disc for this test is from Sergey. So we keep the tissue in 24 hours, and we treat that, and we can send it again into 24 hours. So you can have fresh skin or frozen skin. On this slide, you can have an idea of the results we have in our cutaneous tolerance test. It's classified by product category. You can see first one is the solid swab, the self-tanning products. Again, you have an idea of how many products we find non-irritant and irritant. And all these results are combined in our online application. If you just flash the QR code on the bottom of the slide, you can have access to our demonstration platform, and you can see all the results we have on between different product category. Here you have an example of the skincare products. We found full non-irritant results, but you can with that you can compare the results you obtain on your own product with a global category. You have also the number of step performed, so you can compare it, and it can help you to know if your product is online with the market or not in terms of cutaneous tolerance. This was for the irritation part, and now we work also on sensitization outcome. So this slide is a little bit complicated, but we work under the adverse outcome pathway of the sensitization. again, I will not give you a lot of detail on that. I can speak more if you have any question, but the principle is to divide the outcome in molecular key events. So the 1, the 2, here you have 4 for the sensitization way. And for each key event, some different tests, different in vitro tests correspond to that. And we work under the OECD taste guideline 497 with that. So this guideline defines approaches to combine the results of each test. And with that, you can have an idea of the hazard, sensitizer or not, for your ingredient, or about the potency. You can have the GHS classification, as you see in the bottom of the slide, with a scoring with from the results of these different tests. And I'm going to speak about 2 of these tests. The first one is the HCLAT for human cell line activation test. It corresponds to the key event 3 of the AOP. We can perform it at Eurosafe under the OECD test guideline 442E. It's a specific guideline for the HCLAT, and of course under the combined guideline, the 497. So I'm not going a lot into detail. Just remember that the endpoint is by flow cytometry, and you can have with this test an answer about the hazard and also the potency of your sensitizer. We have a lot of experience with that at Eurosafe, more than 10 years. I will test a lot of ingredients. You have in the bottom of the slide the different type of ingredient. We test a combination of ingredient like extract, complex, or just single molecule, single ingredients. We're trying to develop the keratinescence assay 2 related to the key event 2 of the AOP, again under the 2 corresponding guidelines. So just you to know that next year we will be able to offer the keratinescence assay for you. And we don't only perform tests, we also, as I said, we can also combine the results of the test to give you a specific answer. So with the different results of this test, we're able to combine and to define a sensitization point of departure. So a specific toxicological value about skin sensitization. And with quantitative risk assessment, we're able to give you the maximum concentration of your ingredient that you can put in each product category. You see here at the bottom of the slide an example of what I qualify as what I say the IFRA certificate like. I don't know if you are familiar with that, but it gives you really the maximum level of use you can put your ingredient in each product category. You have for the category 3 for RI products, you can put 1% or with that without any sensitization risk. And we're able to do that with modelization, and we use the thing you say at the right side, the publication from Andreas Knight for that. You have the literature reference also. Back to the safety test, we are also able to perform the senses assay. It's a genomic test, so we analyze directly the gene expression on reconstructed human epidermis. The OECD test guideline is in progress. We expected it for April next year. And as I said, it's a genomic assay. So we look for 3 different group of genes, and when we assess each group of them, we are able to give you a prediction of the hazard and also the potency. The senses correspond to the key event 2 of the AUP, but in the guideline, it might be a standalone essay. We also have a lot of experience with that test. We already test more than 200 compounds, and maybe the main thing you need to remember about the senses test is it's really large domain of application. So every sensitization test, have some drawbacks, because you cannot, some limitation, you cannot test every type of ingredients in this test. For so the S-clad, the deep array, you can not test everything, but with the senses you really can test everything you want to test. So liquid, solid form, the hydrophilic, lipophilic compound, finished product, a medical device by extraction, of course, and spatially soluble and non-soluble test compound. We can really test everything. That is the power of this essay. We also try to adapt this test for finished product here, just to have some confirmation of what we see on the ingredient, because when you add a lot of different ingredients, you can have interaction between them. So with this test, we're able to confirm or not the sensitization potential or the non-sensitization potential. Is currently under development at Eurostat. We presented it at the French Society of Toxicology, and also last September at Eurotox Congress. Now I move forward to the skin absorption studies we can perform at Eurosafe. So we can perform two families of methods. Main common is a static one with the front cells, but we also can perform the dynamic method, and it's really more appropriate to test your product. So here is a classical one under the OECD test guideline 428. You have the classical procedure with the front cells. We use again here skin disc from BIOPREDIC, and with the mass spectrometry analysis, we are able to have the results you can see on the bottom of the slide. So the absorbed dose, but in percentage, and these first two results are used to make margin of safety calculation during the toxicological analysis and assessment. But also with that method, you can have the absorption profile when you check at different time point. But it's complicated with a static method, and this one, the absorption profile is more and more easy with a dynamic method. So we can have two of them at Eurosafe. So this is new thing for us. We use an automated machine here called ReallyGo from the Ex-vivo company. So you put directly your skin disk in the ship, and we can collect after that all the metabolites and all the media going through and passing through the skin disk. So with that really innovative technique, we are able to give you the entire cutaneous passage profile. Where though on the right side you can see the micro perfusion assay. This one is just to find really and to quantify the quantity of available fraction of your product passing through the skin. We just put a little really thin capillary into the skin, and we can collect around time, the available fraction of your product or your active ingredient directly in the skin. So these two methods are new things, and we think that we are able to sorry, I don't find my word on that. So we think that they are growing and growing, and we will see a lot of that in next years from this method. We will see a lot of results from that. Another new thing at WePredict is a transcriptomic approach. So we call it the smartomic RNA-seq. So the principle is to expose a tissue to a product and to perform a transcriptomic analysis. So we quantify a wide range of gene profiles, so up to 15,000 genes. So with that, you can have the whole response of your tissue to your product. So we can compare it to different conditions because it's a quantitative approach. We can compare to, of course, just the media or water of what we want, but you can also compare different products between them 2x2 or perform more complex scientific design. We can perform functional analysis because we can give you results, different results and different pathway activated or not, because we can check the upper regulated genes, down regulated genes. So, and the main thing about this technique is it's really cost effective. So comparing to traditional sequencing things. So at the bottom of the slide, you have just an idea of our protocol, the exposure, the inner extraction, the library preparation, because it's really cost effective because of our specific technique called BRB sequencing. So if you are interested, do not hesitate to ask questions about this new techniques. And I move now to the clinical study because at EuroSafe, we have a really large panel to perform safety and efficacy studies. We have more than 2,000 volunteers. We also have examinators, doctors, and beauty experts for that. In the panel, you can have many different volunteer characteristics. I give you on the slide just an idea of what we can have at EuroSafe, but we can have more than that. So if you want to perform tests, do not hesitate to tell us what you want about that. For safety and efficacy, of course, here you have the patch test, is classical safety test. I'm not giving you more detail here, just a simple methodology to check the biocompatibility of your product and the skin. But more important is the use test under medical control. So as I said, you can assess the tolerance under medical control with all doctors you need. So you can have the claim tolerance tested under dermatological test or ophthalmology is what you want. The idea to test your product on the normal condition of use, because it really represents like this the use of the real consumer. And we can combine that with efficacy study, so with questionnaires, with scoring on volunteers. Here you have the atlas we use to photographic scale by point scale 0 to 10. We also can perform self-assessment of the volunteers with an online tool. I know maybe that point is really interesting for you. We are also able to perform skin microbiota study in vivo and in vitro. We can perform it on our frontier panel. And we work with two of our close partners, Silicium and Sequential, for the sequencing part. But depending on the technique we use, and so, but we're able to deliver results about the skin metabolic test studies. So we're to perform sniff tests here for deodorant and antiperspirant. Also, we work with our partner SenseNet, and it will give you a validity of claims for the deodorant application, 8 hour, 24 hours deodorant efficacy, etc. And to finish with that, you can see on this slide the instrumental measurement and illustrative picture we can perform. So you have here all the different devices we have at Eurosafe for efficacy studying. So the corneometer, the aquaflux, the sabimeter, the glossimeter. Here you have with that an idea of what we can test. The last thing is an illustrative picture. We have a real nice camera for that. So as you can see on the right side of the slide, we can just give you illustrative photo before test and after test to compare to compare different part of the skin. Here you have the face one. So with that slide, I finished my talk, and I'm really happy to take question if you have with that. Thank you for your attention, and I'm waiting for your question.
So good afternoon. It is Yuri Okano from Sora. So today I'm talking from Japan, Osaka. So first of all, I would like to introduce about myself shortly. So I'm Yuri Okano, living in Osaka, and I have been working in cosmetic industry Japan for a long time. So I used to work for the Japanese cosmetic companies for a long time. At that time, so I was in charge of the formulator. So, but I moved to the division of safety and efficacy evaluation, and working for the ingredients company, pharmaceutical manufacturer, and I established my own company. So my company is technical consulting company. So I have some clients in Korea. So thank you for giving me this opportunity to introduce our laboratory, SORA, in France. I will introduce you our laboratory, SORA, and ex-vivo study. So ex-vivo study is unusual study in Asia, but it is very useful. So it is very nice to introduce you about this. So SORA is a contract research laboratory specialized for ex-vivo skin study. So we established SORA 2020 as a collaboration of these three companies. So these two companies is Japanese, and BIOPREDIC, this is French company. So the SORA is located in here, very near from Mont Saint-Michel in France. So in general, we can't obtain excised real human skin easily in Japan and in Korea too. And we cannot carry out the study using such kinds of real skin, especially in industrial research using human skins. But in France, the law is in place, and we can easily get human skin and use these skins for industrial research. Further, BIOPREDIC BIOPREDIC is the biggest provider of prepared human skin discs such as this, and they supply skin materials all over the world. That's the reason why we established SOLA in France. And actually, SOLA is now in the laboratory of BIOPREDIC. So we can obtain very fresh skin materials from BIOPREDIC. So depending on the type of experiments, so sometimes we need fresh skin. In addition, we can obtain a lot of skin discs from the same donor. So it is very useful for the control research. So as you know, control study needs a lot of skin discs. So these are our service. The first one is contract testing. We carried out contract testing for our clients, such as basic dermatological examination or evaluation of raw materials and finished products using the skin discs. And we provide you the open laboratory. You can come to SOLA and carry out your own experiments under professional coaching. Third is we provide training and education of ex-vivo study using the skin discs. So there are the contents of today's talk. The first, I will explain you the positioning of ex-vivo study because ex-vivo study is not popular in Asian area. The next, I will show you the skin tissue from BIOPREDIC. And third, so I will show you some example of the results of efficacy evaluation using human skin and conclusion. So now I will move on the first session. So in the field of evaluation, so there are roughly two categories, in vitro study and in vivo clinical study. Nathan explained about the clinical study in EuroSafe before. So, of course, such kinds of clinical study, the value of the evaluation of clinical study is the highest because the results reflect the real efficacy that consumers feel after using the cosmetics. However, human volunteer cannot be used without safety and quality assurance. Therefore, in screenings or testing of some finished products, so we have to choose in vitro study. In vitro study, so we can use a lot kinds of cells described here. So we can purchase all of them from the provider now. However, recently, many researchers reported that the crosstalk of these all cells are very important for skin reactions. For example, skin is like an orchestra, and keratinocyte is some kind of the violin part. The melanocyte is kind of clarinet part. For white adipose tissues is like a tuba part. So as you can understand, the music cannot understand with a full of parts. So as same as orchestra, so we can't understand the real skin reaction only using only the single cell culture system or keratinocyte or melanocyte and fibroblasts. But in ex-vivo study, skin discs contains all kinds of cells in it, unfortunately without a blood flow. So these are the comparison of transepidermal water loss or between the three-dimensional reconstructed skin and the real human disc. So in the case of three-dimensional reconstructed skin, they have a stratum corneum, but the transepidermal water loss is very high, 40 to 6 grams per square meter per hour, very high. In the case of skin discs, the value is 8 to 15. So this is similar to real human skin. So it means that ex-vivo skin is superior tool with perfect barrier system. So we can evaluate the activity of the test samples, including the percutaneous absorption. So these are the characteristics of in vitro study. There are a lot of strong points. We can screen the ingredients and compare each other. We can investigate the mechanisms of the effects in detail using the biological assay. We can explain the differences between each ingredient, but there are some weak points. We can't evaluate the insoluble materials, sometimes oils. We can't evaluate the finished product because it contains a surfactant or fragrance, such kinds of that. And it cannot reflect percutaneous absorption. So this slide shows the characteristics of clinical study. The strong point is we can obtain the evidences that the sample is effective on human skin, certainly. We can get attractive data for consumers. But the weak point, we can't screen many ingredients. We can't carry out human testing without safety guarantee. And it requires the large scale of volunteers to obtain statistical differences. So finally, I would like to tell you the positioning of ex-vivo study. So this is between the in vitro study and the clinical study. So excised skin is human skin, but we can carry out the biological analysis, and it has a very perfect, very strong, perfect barrier system. So that is, ex-vivo study is very useful tool to connect in vitro study and clinical study. Then I will show you about the skin tissues from BIOPREDIC. So BIOPREDIC has a license to obtain skin tissues from hospitals. The license covers the ethical issue in France. BIOPREDIC processes the skin tissues according to our order and provide them to SORA. So this is BIOPREDIC laboratory, and we have a laboratory on the second floor of the BIOPREDIC. So we can obtain the skin discs very fresh within 24 to 84 hours of surgery. They are very fresh. We can order the conditions of skin discs under these conditions: donor gender, age, ethnicity, such kind of BMI, and anatomic site. But we normally use the abdominal skin and the thickness. We can order the thickness, very thin or thick. Sometimes we can use the full thickness without adipose tissue or with the adipose tissue. So these are the real human skins we can use in SORA. So this thickness is very thin, 400 micrometer. So this thickness is 700 micrometer, and this is full thickness. So it is very easy to treat with materials and also finished products on it. So we can apply the finished products or some ingredients on it, like a cosmetic treatment. Further, we can obtain in the various shapes. The left slide shows the round skin discs. It is very convenient to treat. But some cases that we use the skin sheet. So when we have to evaluate the some beauty instrument, so we use this skin sheet. So this is a brief summary. The data of clinical study is very important, but only the data from outside of the skin can be obtained without the biological analysis. In case of in vitro study, it can investigate the mechanisms of the efficacy, but only it can't evaluate the effects, including percutaneous absorption. So ex-vivo study can observe the response inside the skin, even though it is a real human skin, including the percutaneous absorption of test samples or test finished products. So ex-vivo study is very useful to connect in vitro study and clinical testing. From now, I will introduce you some example of the ex-vivo evaluation. In this slide, I will from the ex-vivo study, so we can obtain the data of gene expression, protein expression, or histological data, or sometimes we can measure the transepidermal water loss or skin hydration using the skin discs. In this slide, I will show you the results of BRB sequencing. So Nathan explained about it as a smartomic sequencing. So BRB sequencing is very good method because we can get many information from the assay. This slide shows the results of BRB sequencing in UVB exposed excised skin. In this assay, over the 10,000 genes were detected, and the expression of 240 genes were different from with UVB or without UVB radiation. And then gene ontology analysis was performed for the different expression genes and picked up the geo term which was higher correlation. For example, so keratinocyte differentiation, and response to reactive oxygen species induced by UVB were picked up, which is very well-known signaling by UVB radiation. Further, cytoplasmic translation and skin development also were picked up as a geo term. So these results gave us not only well-known information, but also new possibilities to clarify the wrinkle formation or pigment formation. This is the results of gene expression with real-time PCR using the skin discs. I showed you the part of the measurement of messenger RNA expression of the epidermal differentiation and the inflammation, extracellular matrix, and the pigmentation, such kinds of that. So this is an example. After UVB radiation, so interleukin 6 expression, messenger RNA expression increasing depend on the dosage of UVB in the skin discs. On the other hand, you can see that this is a collagen synthesis messenger RNA, so UVB decrease the collagen messenger RNA like this. At the same time, BRB sequencing can detect the alteration of wrinkle-related genes. So interleukin-1 alpha, interleukin-6, CXCL8, and MMP1. So UVB radiation increase beautifully such kinds of messenger RNA expression. But in the case of collagen, procollagen, so UVB decrease the expression. This is the results of the testing samples. So in the case of this experiment, we use the testing sample, a kind of plant extract. So we put the plant extract before UV radiation. At that time, you can see interleukin alpha, 6, 8, and MMP1. So increasing by UVB radiation, but after sample treatment, the sample can reduce the upregulation of such kinds of inflammatory cytokines of MMP1. And this sample treatment can increase the collagen synthesis like this. So these results mean the sample may have a ability to improve the wrinkle formation, wrinkle or photoaging. And finally, I will introduce you the histological study using the skin discs. So this is protein expression measuring by histology. This is skin discs, and the blue points shows the nuclear, and the green fluorescence means procollagen, newly synthesized procollagen. So we treat the skin discs. This is no treatment, and this is placebo cream. This is cream containing vitamin C derivative, and this is a cream containing retinol. So you can see a beautiful application of procollagen, green lights here. So it means vitamin C cream or retinol cream onto the stratocorneum can induce the newly synthesized collagen. It is beautiful data. So actually, this is Shiseido's elixir cream. And I will show you the correlation of messenger RNA expression and the protein expression using the skin discs. So this slide shows the vitamin C cream results. You can see that both of them, so messenger RNA level of the procollagen and the protein level of procollagen increase by topical application of vitamin C phosphate magnesium containing cream. So next, I will show you the skin pigmentation induced by UVB radiation. So we irradiated on 3 times onto the skin discs, 3 times UVB radiation onto the skin discs. So after 5 days cultivation from the last irradiation, you can see pigmentation on the skin discs. And this is a Fontana-Masson staining of the skin discs. So you can see a lot of melanins here. So the UVB can induce in the skin discs of the melanin synthesis. So if the treatment of test sample on it, here, 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 and here, so, and the sample, if sample could decrease the pigmentation, it may have an activity of skin whitening. So these are the results of the protein expression in skin tissues induced by UVB radiation. So the upper side is UV radiation, And the lower side, no. So this is no UVB irradiation. The lower side is UVB irradiation. So interleukin-1 alpha is inflammatory cytokines. You can see a very beautiful increase of interleukin-1 alpha. It means the skin discs occur the inflammation by UVB irradiation. And this is MMP1. So MMP1 is very famous collagen degradation enzymes. The UVB radiation can induce the abrogation of MMP1 protein expression in the skin discs. This is collagen. So UVB radiation decreases the collagen synthesis. So you can understand that our skin discs can react to the UV radiation. So next is conclusion. So I would like to summarize my talk. So in ex-vivo study using excised human skin, so we can evaluate ingredients and finished products. So sample can be applied to skin discs just like cosmetic products. And outcomes from ex-vivo study are messenger RNA expression or protein expression by ELISA or histological study. Sometimes we can carry out the Western blotting. And we can observe architecture of alteration of histology and functional evaluation such as barrier functions. So we can evaluate the anti-aging ability or skin lightening ability. Sometimes we can evaluate the anti-rough skin or such kind of that. So this is my last slide, take-home message. So please consider incorporating ex-vivo study as one of your new evaluation method. Thank you for your attention.